The role of IgG Fc receptors in antibody-dependent enhancement. A previously healthy school-aged child has recently returned from the Dominican Republic. A week after arrival, she was rushed to the hospital with fever, headache, abdominal pain, nausea, and hypovolemic shock. An RT-PCR assay was performed and detected dengue virus in her serum, cerebrospinal fluid, and in various organs, including the heart, lungs, and spleen. An immunoassay found high antibody levels for Zika virus components, yet lacked detectable antibodies to dengue virus proteins, suggesting that only a previous Zika infection had occurred. Cross-species neutralization tests were then performed, with low levels for dengue virus suggesting cross-reactivity of Zika antibodies for dengue. Therefore, the previously established Zika antibodies were able to bind to dengue, but failed to neutralize the virus. Instead, their antibody-dependent enhancement had amplified virus entry into phagocytes, promoting viral replication and disease pathogenesis. But what is antibody-dependent enhancement, and what mediatory role do non-neutralizing antibodies play? Antibodies are often viewed as heroes in immune responses, disease-fighting proteins which help ward off foreign substances. This characterization is true of neutralizing antibodies, which bind to foreign targets in a manner which interferes with their function, impairing their ability to enter, fuse, or mature within host cells. Viral epitopes are engaged by variable antigen binding sites in the Fab domain, allowing for opsonization. Antibody-coated pathogens can then engage Fc receptors on immune cells to induce phagocytosis. Fc binding transduces signals within the phagocyte which increase the rate of phagocytic activity, promoting fusion of lysosomes with phagosomes and stimulating virus lysis. However, non-neutralizing antibodies engage targets without obstructing their function, and can engender antibody-dependent enhancement, or ADE for short. In ADE, these pre-existing, poorly neutralizing antibodies enhance disease pathogenesis. They heighten infectivity and virulence by facilitating viral entry and host cell infection. ADE was first reported in dengue virus infection in 1973, with Fc gamma receptors since being linked as key mediators. Fc gamma receptors are surface receptors which bind to the Fc domain of IgG molecules, allowing for internalization of virus-bound IgG and associated infection. Canonical type 1 Fc gamma receptors can either be activating or inhibitory. As humans, we express 4 activating subtypes. These receptors include immunoreceptor tyrosine activating motifs, or ITAMs for short, which are intracellular domains that activate downstream signaling cascades when they are phosphorylated. On the contrary, the sole inhibitory Fc gamma receptor is Fc gamma R2b, which contains an immunoreceptor tyrosine inhibitory motif, or ITIM for short. We also express GPI-anchored FcR3b, which cross-links with other activating receptors to achieve synergistic activation.
In order to understand the effects ADE has on our immune system, it is crucial that we recognize how ADE enters and how it affects the mechanisms of our immune response. To categorize, there are 2 different modes of entry in which ADE viruses can occur: the extrinsic and intrinsic pathway. The extrinsic pathway of entry could be categorized as the antibody-mediated replication of viral cells within phagocytes through entering the cell as a virus immune complex. It is known to enhance receptor interaction rates and therefore results in better internalization and replication of the virus. On the other hand, the intrinsic pathway has been shown to elicit a so-called enhanced immune activation. By a series of reactions, viral antibody complexes inhibit certain interferons that downregulate release of certain cytokines while upregulating others. This causes prolonged infection of macrophages and favors the spread of the virus. The intrinsic pathway that involves immune suppression plays a vital role in manipulating respiratory bursts, the main mechanism of our immune cells for limiting spread and replication of viruses. Respiratory burst is the name of the process in which immune cells such as neutrophils and macrophages produce different reactive oxygen species in response to invading pathogens in order to eliminate them from the host. The production of oxygen and nitric oxide radicals from NADPH oxidase causes oxidative stress on pathogens, while H2O2 hydrogen peroxide is reported to act as a secondary messenger in further intracellular signaling pathways. In non-ADE entry of viruses, respiratory burst is initiated by toll-like receptor 3 and 7, as it recognizes the invading virus that is internalized in endosomes coupled with antibodies. This, along with other receptors, activates the production of interferon gamma and interleukin-8 pro-inflammatory cytokines. Subsequently, the JAK-STAT signaling pathway is activated, and this drives the expression of interferon gamma, interleukin-12, along with nitric oxide radicals, that elicits a Th1 type immune response. In ADE-mediated entry of viruses, however, TLR signaling is inhibited by the expression of TANK and SARM through the virus-antibody complex in non-neutralizing levels. This triggers a different cascade in which anti-inflammatory cytokines interleukin 6 and 10 are produced instead. driving the expression of SOCS3 gene, which then subsequently suppresses JAK-STAT signaling pathway. This, in result, suppresses the production of reactive oxygen species and pro-inflammatory cytokines. And this results in a Th2 type immune response.
Degranulation is a critical cellular process that occurs in granulocytes, such as neutrophils, basophils, and eosinophils, as well as in natural killer cells, upon the activation of Fc gamma R receptors. This process plays a significant role in the immune response against viral infections. So the process of granulation initiates with the activation of Fc gamma R through cross-linking, which is triggered by various signals. And upon this Fc gamma R cross-linking, intracellular signaling pathways are activated, involving kinases like SYK and SRC. The activation of SYK and SRC kinases leads to the assembly of the NADPH-dependent oxidase complex. This complex is assembled in the plasma membrane and phagosomal membranes. This leads to Fc gamma R-mediated PKC activation, which increases intracellular calcium levels, which ultimately trigger the rapid mobilization and release of granule contents. These granule contents include serine proteases, leukotrienes, proteins with antimicrobial activity, and antimicrobial peptides. Now moving on to the next slide, in antibody-dependent enhancement, degranulation is a pivotal process in natural killer cells. And these natural killer cells, specifically upon Fc gamma R3A cross-linking, begin cellular activation and degranulation. The degranulation process in natural killer cells leads to the release of granule contents, including perforin and granzymes. This process is known as antibody-dependent cellular cytotoxicity, or ADCC. The released granule contents induce the formation of pores on the target cell membrane and stimulate apoptotic pathways, ultimately leading to the death of the infected cells. These immune mechanisms involving degranulation in granulocytes and natural killer cells collectively contribute to efficient elimination of infected cells.
During an infection, pathogens interact with antibodies produced by B cells during an adaptive immune response. The Fc domain of these antibodies interact with the Fc gamma receptor, which results in cross-linking. Cross-linking is a process that facilitates signal transduction, which ultimately drives the phagocytosis of pathogens. This study by Louis John shows that when you knock down Fc gamma receptors, you suppress antibody-dependent enhancement activity, which confirms the role these receptors play in antibody-dependent enhancement. Furthermore, the phagosomes then fuse with the lysosomes traffic from the cell secretion system, where they can be degraded to attenuate infection. This mostly occurs in cells like macrophages. Then, an MHC-class II molecule is trafficked into the phagolysosome with its peptide binding cleft facing the cytosolic side. The degraded peptide is then placed into the peptide binding cleft for antigen presentation on dendritic cells. The MHC-class II molecules are then presented on the cell surface of antigen-presenting cells like dendritic cells to facilitate adaptive immune responses.
Again, as previously mentioned, ADE can trigger other life-threatening diseases such as HIV through its projection of IgG antibodies. Many studies have been performed where it became evident that HIV infection is highly reliant on FCYR interaction once the monoclonal antibodies fighting against HIV, also referred to as anti-HIV MABs, are introduced within the mice samples of direct HIV virus-provoking infection. The ultimate purpose of anti-HIV MABs is to take preventative measures and trigger protection. It's important to note the difference between neutralizing and non-neutralizing antibodies. Neutralizing antibodies, specifically MABs, can trigger the spike protein, which binds effortlessly and prevents further interaction or entrance of virus as it approaches the host cells and battles. Non-neutralizing antibodies are not specifically warding off the infection. Instead, they supply regular antibody effort in contribution to maintain a systematically working immune system. It's evident that a trial that involved RV144 HIV vaccination presented interaction with FCYRs that stimulate Fc effector functions is highly correlated to the presence of non-neutralizing IgG antibodies, revealing that Fc and FCYR communication strains protection against HIV-infected individuals, and raises the idea that ADE vacancy is enhanced once the progression of FCYR and Fc begin. In conclusion, FCYR and its diverse list of receptors are the key mechanism in the assimilation of pathogens that leads to such lethal diseases, which continue to deteriorate the immune system further. Studying the pathway and correlation and importance of MABs can lead to diminishing viruses and specifically engineering proper binding to ensure sufficient potency and clear results of efficacy.
Future approach of Fc receptor in SARS-CoV-2 research. Variants of Fc gamma R pathway provide insight for antibody-mediated protection against SARS-CoV-2, lead to the development of effective vaccine and MABs-based therapeutic strategies. Fc gamma R engagement on dendritic cells can be used to stimulate cytotoxic antiviral CD8 and T cell response, especially during severe SARS-CoV-2 infection. Biologically relevant in vivo model of Fc gamma R can address the inadequacy of current in vivo control system in replicating the intricacies of SARS-CoV-2 infection. Also, selective engagement of specific activating Fc gamma Rs on this distinct leukocyte type is used to aim for rapid clearance of obsolete virulence and cytotoxic elimination of infected cells to control viral replication and tissue damage.